Hello all, and welcome to this lecture on B cell development and maturation. As you likely remember from prior lectures, all the figures that come out of the Janeway textbook will be referenced on the slide by number in the upper corner or lower corner of the slide so that you can follow along in your textbook as we go. This will be particularly useful for any of the figures that have small writing that are hard to see on the video. So I'd like to start out the lecture with some primary objectives. These are the things that we hope to learn over the course of the lecture. So by the end of the lecture, we should know each of these primary objectives. The first one being knowing the stages of B cell development, and this all occurs within the bone marrow. And then we will also talk about the steps of VDJ recombination, not on a mechanistic level like was done in the lymphocyte antigen receptor lecture, but more relating it to the different stages of B cell development when they occur. And then the second primary objective is learning about the process called allelic exclusion, and this is a way to enforce a single B cell receptor for every B cell that there is. So just in terms of the immunological response, during the adaptive immune response, we only want one B cell receptor per B cell, and this is enforced by allelic exclusion, which we will talk about later. And then the final primary objective is learning about B cell negative selection, which is a way to select against B cells that have autoreactive or self-reactive receptors. So these are receptors that can interact with the self, and if they are allowed to persist, this could result in autoimmunity. So an important thing to remember is that lymphocyte antigen receptors are different from innate antigen receptors in that they are specific and unique. So in our lecture about lymphocyte antigen receptors, we have done this compare and contrast before, but just to reorient ourselves, innate immune receptors bind to general features of pathogens so that they can recognize a broad array of different pathogens. And this allows them to recognize a finite number of pathogen-associated molecular patterns, or PAMPs. By contrast, the lymphocyte antigen receptors are unique on B and T cell receptors, and they are specific, and they theoretically could interact with any molecular pattern on a particular pathogen. So these are usually specific to a singular pathogen as opposed to providing a broad immunological protection. Now that we have reviewed the differences between innate receptors and lymphocyte antigen receptors, I want to move back to B cell development, and this is going through the stages at which these B cell receptors will be produced. So just giving a general overview of B cell development, and this is a figure in particular in which it might be useful to follow along in your book because the writing is a little bit small, but I just want to highlight that we start in the bone marrow as a hematopoietic stem cell, and this will give rise to all the blood cells, both the myeloid lineage and the lymphoid lineage. So moving on during differentiation, the hematopoietic stem cell will move into this common lymphoid progenitor stage during differentiation, which will give rise to many different cell types, including B and T cells. But in this particular lecture, we are particularly interested in focusing on B cells. So after developing in the bone marrow, these B cells will move into the blood and the secondary lymphoid organs. And then eventually these B cells will take on important roles in the immune system called their effector functions, but this will be primarily covered in subsequent lectures. Today, we want to focus primarily on the development of the hematopoietic stem cell moving through the stages of development in the bone marrow to when they exit into the bloodstream as mature B cells. So just to contextualize this development a little bit more, going through the life cycle of the B cell will start out in the bone marrow, which is where all of the development occurs. And then they will move on to their mature stage, where they move into the secondary lymphoid organs. And then, as mentioned previously, during the immune response, they will move into this effector stage in which they can elicit an immune response, and this will be primarily covered in subsequent lectures about B cell activation and response to different pathogens. So before we jump into B cell development, I want to talk about the major goals that we hope to accomplish during this developmental process. So the first is the rearrangement of the heavy chain. And the mechanism by which this occurs is VDJ recombination, which, if you remember back to our lymphocyte antigen receptor lecture, that we covered how this mechanistically occurs, and we hope to contextualize this in the context of B cell development here. Importantly, then, the rearrangement of the light chain for forming the antigen receptor, the B cell receptor, will occur. And the B cell receptors have two options for their light chain, the kappa or the lambda chain, and we'll get into how this specifically occurs. And then lastly, we want to eliminate or select against B cells that have autoreactive receptors. So these are ones that could stimulate autoimmunity in the host because they react to host proteins or carbohydrates or etc. So moving into an overview of the different stages of B cell development, which we will go through each stage more in depth, but this is just a general overview. We start with our stem cell, which are the progenitor cells for all blood cells. Then we move into the phase called the Progenitor B Cell phase, in which the rearrangement of the heavy chain will occur, and this will happen in a sequential order. And then after that, we move into the pre-B cell stage, in which the rearrangement of the light chain will occur, which also happens sequentially. And then lastly, we have elimination or selection against the autoreactive B cells in the immature B cell stage before they mature and are able to circulate in the secondary lymphoid organs. So moving specifically through each stage, we start out as this hematopoietic stem cell, which is a pluripotent cell, meaning that it can give rise to many different cell types. As it does, it gives rise to all the different blood cell types. And it's self-renewing, meaning that it will give rise to other stem cells that will continue to propagate and produce more blood cells. Then this stem cell will transfer through the common lymphoid progenitor stage, and during this stage specifically, this is when the B cell will commit to a B cell lineage, or this is when the blood cell will commit to a B cell lineage. And this is driven by B cell specific transcription factors that allow for differentiation and commitment to this B cell lineage. One of the important proteins that is upregulated during this particular stage of development and is stimulated by these B cell specific transcription factors are RAG proteins, which, if you remember from our lymphocyte antigen receptor lecture previously, that these RAG proteins are necessary to catalyze VDJ recombination, which is the recombination events that results in these unique lymphocyte antigen receptors. So during this pro-B cell stage or progenitor B cell stage, we start out with the D to J segment rearrangement. So here are our two alleles of our heavy chain that are present in the B cells, which consists of variable diversity and junction segments, as we have covered in the lymphocyte antigen receptor lecture when we went through the specific mechanisms by which RAG catalyzes the recombination events. And so the first stage is the recombination of the D to J segments, which will happen at both alleles. So here I'm showing both alleles in which the D to J has been joined together. And at this particular stage in the progenitor B cell, no protein will be expressed on the surface of the B cell because we do not have a full protein that is encoded. We just have the D and J segment joined, and it is not until the V segment is joined that we have a full heavy chain that can actually be expressed as a protein. And that's just highlighted here. No functional B cell receptor is yet expressed. So moving to the next stage, from the early progenitor B cell to the late progenitor B cell, we have the V segment of the heavy chain joined to the DJ segment of the heavy chain. The important distinction here in this particular recombination event is that it happens at one allele first, and then it will move on to subsequent alleles in the case of failure to recombine successfully. So demonstrated here, we have our D to J segment joined on allele 1, and then recombination will attempt to occur. And if this recombination event is successful, then we will have this process called allelic exclusion, which is the process by which the B cell will suppress epigenetically the recombination at the second allele. And this is a way to enforce one B cell receptor per B cell to be expressed. And so then I've talked previously about this idea of successful rearrangement, but we want to get into what that really means. So this means that the rearrangement results in a protein that is in frame, which means that there are no premature stop codons, and that you can have a fully functioning protein that still has the same structural features. And so this will result in expression of the protein on the cell surface that is capable of signaling to the cell in the proper way during an immune response. So moving back to our allele 1, if the recombination event is unsuccessful, meaning that the recombination is out of frame, then this allele will have another chance to undergo rearrangement called successive rearrangements. And the reason that this allele is able to do so is because there are multiple V segments under which recombination can occur. If we remember from our lymphocyte antigen receptor loci lecture, that there are many V, Ds, and Js, so as long as there are still Vs to recombine with the DJ segments, successive rearrangements can happen at this allele, trying to undergo and have functional rearrangement at the heavy chain. After these successive rearrangements, if we have successful rearrangement, then the cell is able to move on to the next stage of development. And in the case that this is not successful, so if the allele, the first allele that underwent recombination, if this fails, then recombination can try to be attempted at the second allele. And if this is successful, then you have a proper heavy chain and the B cell can move on to the next stage of development. In the case that this doesn't work, then there is no heavy chain that can be expressed by the B cell, and then the B cell will die at this stage of development because it cannot produce a B cell receptor, which is necessary in its stages of development. So in the event that we do have a successful heavy chain, or that we don't, how do we actually test for a successful heavy chain? And the way that this is brought about is by mimicking B cell receptor signaling. So if we look at our B cell receptor here, when antigen is recognized by that B cell receptor, there are several adapters that result in downstream signaling within the B cell. And these will be covered more in depth in the B cell activation lecture. But in this particular instance, we can mimic this interaction of an antigen with its particular receptor as a way to signal to the B cell that we have a B cell receptor expressed on the surface of the B cell successfully. Something that's important to keep in mind here is that for a B cell receptor to be expressed on the cell surface, we need to have interaction of a heavy chain, which we've already gone through the recombination events for, as well as a light chain. And this pairing of the heavy chain and the light chain is necessary for the B cell receptor protein to make it from the endoplasmic reticulum and to be exported to the cell surface. And without that, we have other ER retention factors or endoplasmic reticulum retention factors that prevent the heavy chain from making it to the cell surface by itself. And this is really just a matter of stability, because the protein is unstable without being paired with a light chain. Now, as many of you may have realized at this particular point in time in the B cell development, we do not actually have a light chain that can allow for cell surface expression of a viable BCR because light chain rearrangement hasn't occurred yet. And so how do we overcome this problem of not yet having a light chain receptor or light chain part of the receptor? So one of the ways that the B cell overcomes this problem is by expression of a surrogate light chain. And so this kind of takes the place of the light chain and allows for secretion from the ER to be expressed on the cell surface of the B cell, just taking its place. And this consists of this V pre-B segment as well as this lambda 5 segment shown in blue and orange on this diagram. And if we zoom in to the particular structure of the surrogate light chain, the way that we mimic antigen recognition and allow for downstream B cell receptor signaling is by cross-linking of these receptors, because the surrogate light chains have these regions that are able to interact with each other, and because they can interact with each other, they allow for the adapters that interact with the intracellular portion of the B cell to come together and trigger this downstream signaling. And these downstream signaling events will become more apparent when we learn more about these adapters, particularly during the B cell activation lecture, but the process by which this occurs mimics that of antigen recognition. And so once we have a heavy chain paired with a surrogate light chain expressed on the cell surface that is able to undergo and initiate downstream BCR signaling, this tells the cell that there's successful rearrangement of the B cell receptor. And it also gives the B cell a signal to proliferate, much as the antigen recognition signal during B cell activation during an immune response also allows for the B cell to proliferate. So just as a summary on our path to heavy chain rearrangement, we start out with our pro-B cell. And then as the B cell develops, it will start to upregulate RAG expression, which is necessary to catalyze VDJ recombination. Then we start recombination of our D to J heavy chain segments on both alleles. Then the recombination between the V to DJ at one allele will proceed. At this point, it's happening sequentially, but not at both alleles. And in the event that we have a successful or in-frame rearrangement that allows for expression of the heavy chain in combination with the surrogate light chain on the cell surface, this cell will be able to move on to the next stage of development. But in the case of a recombination event that results in a protein that is out of frame, which will occur 2/3 of the time, then the cell will have to undergo additional recombination events at the second allele. And so if the event is successful, I realize I forgot to say something on this particular slide. Can I just start over for this slide?
The entire slide, or just pick up from where you are?
I forgot to mention allelic exclusion at this particular point.
Okay, so just pick it up from there.
Okay. So when this rearrangement is successful, this is where we have allelic exclusion. So this will suppress recombination at the second allele. And if you remember to what we talked about previously, this is necessary for there to be only one B cell receptor expressed on the B cell surface, which is necessary for its proper immunological function. So in the event that this recombination event is unsuccessful, then the B cell will proceed with recombination at its second allele. And if this is successful, once again, the B cell will be able to move on to subsequent stages of development. And this is tested once again with that pre-B cell receptor that consists of the surrogate light chain and the heavy chain being expressed on the cell surface. At this point, if heavy chain rearrangement is unsuccessful at the second allele, then the B cell will die because there are no more opportunities for heavy chain rearrangement.